Assalamu alaikum wa rahmatullah. I'm very happy today to be here in this thesis defense of Mrs. مريم العمارتي in the presence of first the reporters, Professor Rashid Jawdi from Faculty of Medicine and Pharmacy, Mohammed V University at Rabat, and then Professor Leila Sbabu from the Faculty of Science, Mohammed V University in Rabat as well, as a reporter. And then Professor Mohammed Azami Idrisi is from the Faculty of Medicine and Pharmacy in Faz, from the University Sidi Mohammed bin Abdullah in Faz. He is with us online. He is not present physically with us. And then we have as examiner Professor Lahsan Bilyamani from the Faculty of Medicine and Pharmacy, Mohammed V University as well, at Rabat. And then Professor Mona Boudgheri from the Faculty of Medicine and Pharmacy, Mohammed V University of Rabat as examiner as well. So the title of the presentation is Mycobacterium tuberculosis and SARS-CoV-2 genomic analysis, input into outbreaks and surveillance investigations. So, Mrs. مريم العمارتي, you have about, we're going to listen to you first. So we have about 35 to 45 minutes to tell us about your work. Good luck to you. And you have the floor.
Thank you. So, thank you for your kind introduction. And I'm very grateful for the committee member and also the audience for being accepting and coming to assist my thesis. So I'm glad to be able to present my work on Mycobacterium tuberculosis and SARS-CoV-2 genomic analysis, and how we can use genomic as input to have an input into outbreaks and surveillance investigations. So as we have all experienced during the last year, infectious diseases can cause pandemic, which have a serious potential to overwhelm the capacity of any country with serious health and economic consequences. So an infectious disease is a disease that is able to spread either directly or indirectly from one person to another or from animal to people, which we call spillovers. They are mainly caused by pathogenic microorganisms such as bacteria, viruses, parasites, or fungi. We can think of the spread of infectious diseases as a continuum with three different stages. At the low end of the continuum, we had the endemic phase, when the number of the diseases is under control. However, this number is going to be different depending on the disease. For example, if we take the example of the flu, we'll be expecting several cases here and there at the same time in the same place. And it should be normal without any alarming bells. However, if we're talking about the smallpox, which have been eradicated years ago, there is no normal number. Even one case would be considered too much, or even the start of an epidemic. So according to the Health Organization, an epidemic is when the disease moves from being a problem affecting few people here and there to something that have an impact on the community, as it was the case of tuberculosis and leishmaniasis years ago in Morocco. However, when we talk about pandemic, is when the disease moves from being a community problem to something that have an impact on the world, a global threat, like the COVID-19. So if we take a look globally at infectious diseases, we can see that they constitute 26% of the global burden of diseases, causing more than 14% of the death. So recently, the Health Organization has published a new research blueprint priority list for research and development, and which include mainly respiratory infectious diseases. So we'll be using two examples from the list in the following slide as case studies. So the first example is the COVID-19, which is as a response for the current situation, and tuberculosis, which have been in the last 4 years as a plan from the Health Organization to eradicate TB by the end of 2030. So the COVID-19 pandemic started by few cases in Wuhan, China, and spread few months later to the rest of the world. It's caused by a virus belonging to the coronavirus family, which normally causes respiratory diseases, or what we call the common cold. This family also has a history in epidemics, such as emergence of MERS and SARS-CoV-2 years ago, SARS-CoV-1 years ago. If they are only responsible for 171,000 million infections and more than 3.5 million deaths. So if we take a global look at the structure of the virus, we can see that it is a simple virus. However, in a piece of RNA that's called for 13 protein, the main protein is the spike protein, which allows the virus to bind to the protein and ACO2 protein at the surface of the host cell, allowing the cell entry. So there is no treatment for the COVID-19. However, different vaccines have been developed, and many countries have already started their vaccination campaign. So if we talk about tuberculosis, which is the major killer surpassing HIV and malaria, it is the most successful human pathogen. It takes weeks to develop and months of multi-drug therapy to cure. According to the Health Organization report in 2020, we can say that tuberculosis has infected more than 10 million people and caused the death of more than 1.4 million people. But in reality, there is 2 billion people who are infected with TB at latent form, risking from 5 to 15% chances of reactivation at some point of their lives. So the TB is mainly due to a mycobacterial tuberculosis bacteria that primarily affect the lungs by escaping the immune system. So if diagnosed properly, the TB tuberculosis can be treated within 6 months using a 4-drug therapy. However, things are not that simple. There is emergence of MDR TBs have complicated things, and also the limited resources for the second-line chemotherapy, which is a very heavy treatment that can last 2 years, using expensive, toxic, and not so much available drugs. We have also the emergence of the new TDR, which are totally drug resistant, meaning there is no antibiotic that can have an impact on them. What makes TB hard to eradicate is mainly it's influenced by other factors, the emergence of new epidemic. The HIV epidemic in the 80s have a very strong boost on the emergence of multi-drug resistance and TB in general. Also, the breaking down of the national control programs that happen very frequently. We have also the migration from countries with high burden to countries with even not a program, a BCG program vaccination. And lastly, we have right now the COVID-19, which will have huge consequences in TB in years to come. So recently, a new field has emerged, genomic epidemiology, which uses sequencing to get an insight into the genome of pathogens, making it easier to detect pathogen even before an outbreak or during an outbreak. We can also track this pathogen during an outbreak doing transmission routes or following variation emergence. It's also used, as it was the case in Morocco, in decision making, and also as the first step in diagnosis, treatment, and vaccines. All this was possible in large part by the fantastic advances that happened in the sequencing technology, and also by the lowering of the cost of the sequencing. So the main objective of this thesis is to use genomic epidemiology as a tool to help during outbreaks and manage better pandemics. By answering this question, what's called infection, where is the infection agent is coming from, and how fast can the pathogen spread? What's the impact of mutation in the affected agent? So to do so, I will be using COVID-19 and tuberculosis as examples to get insight into each of these questions. So we will start by the first question, what's causing the infection? And we will answer actually this question by accident when we received 11 strains from mycobacterial tuberculosis from the military hospital here in Rabat. We received them as belonging to the mycobacterium tuberculosis complex using high genotype. So this technique allowed the detection of the TB complex as well as the first and second line drug. Once we have done the sequencing, we follow up with the mapping of the genomes to the reference genome of TB. And what we found, 2 out of 11 strains didn't map to the reference genome, with showing a very low percentage of mapping between 5 to 7, which was very abnormal for 2 strains belonging to the same species. So what we did is we did an assembly of the TBs and tried to check with a quick blast. The results show that both strains belong to Mycobacterium tuberculosis with 100% identity. So recently, our collaborator at the military hospital have published a new paper comparing the specificity of Hain comparing to the TB culture, and they found that Hain have a 4% error positive, false positive. So besides the accuracy of the whole genome sequencing, the sequencing can also save time. You can do in weeks or less than a week what it takes you 15 weeks in diagnosis TB. It also will improve treatment accuracy as well as output. Also, you can identify co-infection which is very common in infection with TB. And the wrong assumption that we have on tuberculosis and whole genome tuberculosis, whole genome sequencing of TB, is that it's expensive. However, the Public Health England have demonstrated that the sequencing of TB is faster and much less expensive than using the 15-week with standard diagnosis workflow. So during an outbreak or an epidemic, it is very important to identify the origin of an infection in order to limit the spread. In TB, for a better assessment of the relatedness between the TBs or the strains, we use the classical genotyping technique, such as polytyping or mirror-VNTR, which normally cover 1% of the genome. However, if we use the whole genome sequencing, we can stretch this coverage to 90%. Providing an added resolution for examining the genetic relatedness, as well as capturing much more genetic changes that can occur. And we have done that in the 9 strain that we have obtained sequenced. So we retrieve all the SNPs and variation that we have in the 9 strains, as well as the variation that of 70 strains that we extracted from the database. And we generated SNP phylogenetic tree. So the results show that there is a low lineage diversity among the 9 strains that we have, since all of them belong to the lineage 4. However, polytyping analysis show that there is 2 main sublineages: the LAM, which is Latin American Mediterranean, and Harlem. And if we take a look at the Harlem sublineage, we can see that we have 2 strains, MTB1 and MTB2, that have the exact same polytype. However, the phylogeny, if we look at the phylogeny, we can see closely. Can I see here? So if you look, we can see that MTB3 is much more diverse than MTB2. Maybe MTB3 has emerged from MTB2. Maybe there is a spread of the same plot in some location in Morocco. So, but we cannot answer this question due to the lack of epidemiological data. This lineage, the lineage 4, has a very good prominent presence in Morocco, in North Africa in general, as it was previously described in Morocco, Egypt, and also in Algeria. So suggesting that maybe the immigration from Europe to Morocco in the last 30 years was behind this prevalence. We can even go further and try to analyze the number and identify the number of entries of the pathogen in a population. And we will take the example of SARS-CoV-2 introduction in Morocco. So number of entries means if the transmission is due to one clone or one strain, or there is multiple strain transmissions. So to do that, we started by doing the sequencing of 15 strains of SARS-CoV-2 using MENINE and combine them one strain from Morocco from the GSA database. Also, we tried other strains from different parts of the world and combine all of them to trace this phylogenetic tree. So the tree will give us idea about to which lineage these strains are belonging and to which country they are most closely to. But we cannot give much of the more details. So all the strains in Morocco are belonging to the lineage G, which is European lineage, harboring the mutation 614. If we take a look at the haplotype transmission network that we draw using phylogenetic tree in combination with epidemiological data, we can see that we have 4 main haplotypes. Meaning, in Morocco, there have been 4 different entries of SARS-CoV-2 in Morocco. And also, 19% of infection are resulted of transmission within Morocco and not from the outside. So it is important to note that phylogeny contains information not only about individual cases that we have sampled, but also cases that we have not directly observed, such as the ancestor, and will enable us to do a molecular dating to establish the date of starting of an epidemic. So we will take the case of the COVID-19 pandemic using our study of 3,000 genomes. And when we did the phylogenetic tree using molecular dating, we observed that actually the outbreak started in Wuhan, China, between October and November with 100% confidence. This means that the virus had been circulating for 2 months without being detected in Wuhan. We can do the same thing for tracking new variants, such as the variant of 614 that was known to be a European variant just because it was detected for the first time in Germany. However, our analysis of genomes from December to March showed us that this genome, this variant, in fact, was first detected in Shanghai, Asia, but didn't gain any predominance. However, it gets its predominance once it left territory. So this means that maybe in China, the genetic variation, the genetic of the population and the environment was different than the genetic and the environment in Europe, which enabled the virus to spread much more. The genomic epidemiology also can enable us to trace transmission routes, and we will do that with the COVID-19 study that in which we analyzed more than 3,000 genomes. So we traced this map or animation using the next training package and in-house script. And as we can see here, all Asian strains diverged and resulted in other strains in all investigated regions. However, we can see that European strains give birth to the strains in Americas and also Africa, and also multiple strains within Europe itself. This divergent strain came back as new strains to Europe and also Asia. We can do similar animation and transmission routes with Moroccan strains. However, we are facing the same problem. We have the lack of epidemiological data, and also the majority of strains that we are sequencing are from the same origin, Rabat or Casablanca. So we don't have much power to have an accurate transmission route. In general, pathogens accumulate genetic changes at the same time scale of which the transmission is occurring in an outbreak. It was observed in TB, in TB transmission and diversity, when we try to analyze the number of variation, which include SNP, deletion, and insertion. So we called all this variation, and with in-house scripts, we tried to identify all the shared mutation between these strains. And as we can see here, all these strains have shared almost 400 mutations. However, which is similar to the Harlem group, which also share more than 400. However, if we take a look at the Lum strain, we can say that the number of shared mutation is 100. So there is a high mutation rate of the Lum line compared to the Harlem, which may be because the genome of the Lum line are hypervariable. And we will see that in different results that we will see later in the presentation. So more than half of the insertion and deletion and mutation that we have detected were present in the PPE genes, representing a major source of variability for TB. These genes also contain repeated regions, which make it hard to sequence with short reads. So we cannot have a complete genome using short reads for TB. So the accumulation of mutation in general depends on the pathogen. The SARS-CoV-2 is a very good example to study the accumulation of mutation. And we have done that using our first study in 3,000 genomes. And as we can see, the mutations have accumulated at a relatively constant rate from the start of the outbreak till March, with 11 mutations per genome. We also did the divergence tree, which allowed us to estimate the mutational route of the pathogen. And as you can see here, COVID-19 or the SARS-CoV-2 has a mutational rate of 0.008 mutation substitution per site per year. I'm sure that this number has changed in the late month of the pandemic, since this study was in the first month of starting the pandemic. However, the divergence is mostly affected by the host genetic and environmental factor, since we consider it as a mechanism for the pathogens to find the right fitness profile to survive and thrive. So we have tried to analyze the divergence within the genomes and also the divergence within each continent and each country. And we started by assessing the divergence of each genome compared to different genome from Wuhan. And also, what we found, we tried to estimate the number of divergences or the rate of the divergence in each country using this formula. So what we found is that the major divergent countries were Serbia and Hong Kong. But if we take a closer look to Asia and Europe, we can say that European countries are much less divergent than the Asian. This high diversity in Asia could be due to the multiple sources of interaction of the start of the epidemic. However, the low diversity could be due to the early introduction and rapid spread that happened in the Europe, and also the genetically close variant that originated from the original strain. So as we have all seen that Europe during the first month have witnessed a very rapid spread, which means we have a single source leading to multiple infection. So the virus will not have much of time to stay inside the host to have multiple cycles that allow it to change. Therefore, we have lower divergence. This divergence accumulates mutations. So when we analyzed the 3,000 genomes, we found that we have 3,000 mutations that we have presented here in the genome of SARS-CoV-2, and all majority of this mutation we present at this gene, which is OFA1. So if we take a look at how many mutations have a prevalence which is superior to 1%, we can find only 169 mutations, which means 0.5%. And when we take a look at the number of mutations that have which consider hotspot mutation, we have with a prevalence superior than 10%, we can find only 14 mutations. So these hotspot mutations show different profile according to the geographic regions, and also we can find mutation in some geographic region and not in the other. So this difference in mutation is mainly due to pressure selection made imposed by genetic of the host or the environment. And to calculate this selective pressure and study it, we have used this method of studying the ratio between the non-synonymous mutation per synonymous mutation. And what we found is that the spike protein and OF1AB protein genes are under negative selection. And also, we found multiple sites under negative selection. 6 sites in the spike protein and 8 in the OFAB protein, while only 1 site has been found in each of the genes. So, however, we cannot take this result seriously because the sampling size and the early date of collection were very, due to sampling size and the early date of collection. So, none of the hotspot mutation was identified because they didn't emerge yet. The study was performed in a collection between December and March. So the negatively selected site could indicate a functional constraint and could be useful for drug and vaccine targets since they are conserved and therefore less likely to change. This type of selection pressure induced mainly as a result of changing environments, such as the case of emergence of anti-drug resistance as a response to the use of antibiotics or misuse of antibiotics. The TB is an excellent case for the depression by the antibiotics posed on the selection of TBs. So we can find 3 types of TBs. MDR, which are resistant to at least both isoniazid and rifampicin. XDR-TB, which show resistance to isoniazid and rifampicin and also quinolones, and at least one of the second and the 3 second-line injectable drugs. We have also the emergence of the TDR, which is resistant to all the drugs that we have. So here we try to compare the analysis or the results with the genotype and also the results of resistance using the sequencing and combine them with the phenotypic test in order to have a clear idea about the resistance of TB. So before we start analyzing, we can say that just by using the Chaîne genotype results, we can say that we have 2 clusters, the Harlem and the Latin American Mediterranean. All the strains that have a mutation in LPOB, KTG, or INHA, confirmed resistance to isoniazid or rifampicin, belong to the Latin American Mediterranean clade, which explain why this clade or why the lump clade was more diverse and have less mutation share between his strains. This previously was described as a common phenomena in resistant TBs or resistant strains that have to do multiple mutation in order to have some fitness, because the resistance normally come with a cost for a bacteria. So the approach that we use in the sequencing and obtaining resistance from sequencing is we try to retrieve all the genes associated with resistance from the database, and also retrieve all the mutations that have been confirmed to be associated with resistance. and created a database that we use as a reference in order to retrieve all the mutations that can have an impact on the resistance. So we found much more mutation than the hind can have found, but this mutation, however, cannot explain all the phenotypic resistance results. If we take an example for the cyclosamine, which is resistant here to all in all the strains, there is none of this mutation can confer this resistance. So we can say maybe there is another mechanism that confer resistance outside of mutations. Maybe there's another gene. Maybe there is pump efflux that can induce the resistance to cyclosamine. However, to confirm this result, we don't need to do sequencing analysis but transcriptional analysis to see if there's any expression of this gene while the bacteria is under pressure of antibiotic. We have also witnessed this with moxifloxacin, which is quinolone antibiotic in the second-line treatment. So in order to try to answer this question, we tried all the mutations that we found in the resistant-associated genes and were not previously described in the literature, in order to see if any of these mutations can confer resistance to the bacteria. So, and what we did is we retrieved the protein, multiple protein, candidate protein like KTG and LPOB and GAB complex, and we tried to introduce this mutation. For example, if I take the example of the LPOB here, we retrieve the genes and try to insert this mutation and see if there's the binding affinity with the ligand, which is rifampicin, will decrease or increase. So what we saw here is there is a decrease of the binding affinity, meaning that the antibiotic doesn't have much effect on the protein anymore. There is a slight increase of resistance. And we can see this much in a much clearer with the gyrase AB complex, which show that there is a resistance to moxifloxacin and levofloxacin if the strains have this mutation, the 439. So we did similar thing to analyze the effect of the 614 mutation in the SARS-CoV-2. And here at the left we can say you see the wild type of the spike protein. You can see there is two hydrogen bonds that link the subunit 1 to the subunit 2. However, when we introduce this mutation, these two bonds have disappeared. So the loss of these two hydrogen bonds between subunit 1 and the subunit 2 in the spike protein increased the rates of the cleavage. Therefore, improve the virus entry to the host cell, as it was previously described. So we have tried to enlarge our analysis and include many mutations that we find in the 3,000 genome analysis that we perform. And we tried to look if mutation at the RBD of the spike protein have an impact on binding ability with the ACE2. And in fact, what we found is there is 4 mutations that potentially increase the binding affinity of this of the spike protein with the ACE2, which means maybe in the future they could have an impact in increasing the spread of the virus. So lastly, the incorporation of pathogen genomics into reporting and policy making is one of the great things that we should do during the pandemic. One of the biggest problems in genomics was what happened to the sequencing when they are generated. The health organization advocated that pathogen genome sequencing, especially during public health crisis, should be shared immediately after generation. So Morocco have tried this same protocol and start with the National Genomic Surveillance Program. There is a consortium that have been made between many institutions trying to sequence as many strains as we can. So our lab sequenced more than 200 genome sequence of SARS-CoV-2 using many technologies such as the nanopore, economictic, and ion-tolerant S5. We have also tried to build a database at the beginning of the pandemic in which we put all the strains of the COVID-19 data in this period from the NCBI and from GSAID. We tried including the analysis of these variants so the user can check the variants and also compare them if they have compare them to their results if they have data. And recently we have also added a pipeline that allow the user to analyze, especially non-bioinformatic people, to analyze directly. So we just put your FASTQ file, raw FASTQ file here, and press the button to get the results of the PUM if you want to do a visualization, consensus sequence if you want to do phylogenetic tree, or VCF annotation if you want to have an idea about the mutations that are emerging. So in conclusion, genomics can be a powerful tool to predict drug resistance, diversity, transmission, and also predicts the effect of mutation on pathogen evolution. So in Morocco, what we should be focusing on in including the whole genome sequencing in diagnosis, especially for TB diagnosis. Also the generation of data and centralization. All institutions should share their data and keep them. So in the next outbreak or next epidemic, we can have a reference to which we can go back to. Also performing deep sequencing, which means sequencing all of the multiple pathogens at once when we don't know what's the cause of infection, is a very important approach. So because one thing is for sure, pandemic pathogens will strike again. However, before I finish, I would like to thank all the people that helped me during my journey of PhD, starting by my supervisors, Professor Ibrahim Yazdi, which I know for 13 years now, and thank you for your help, your guidance, and also for all being patient with me sometimes. And I would like to thank also Professor Samir Siak for his kindness and positivity. The committee member, I'm very grateful for your time and that you took to read my manuscript, especially during the month of Ramadan. And of course, I'm very grateful to our collaborators in the military hospital as well as the University Mohammed VI of Science and Health, with which we have worked there in tuberculosis, also SARS-CoV-2. To all the lab team, there's a big list, so I cannot include all the names, but I'm very grateful for all the time that we have spent together. And lastly, my family and friend, thank you very much.